Hello and welcome back. This is the second segment of the digestive system lecture. And in this second segment, we will cover bullet point 3. Let me highlight bullet point 3. We will look at the processes that occur as food material travels through the GI tract. So we'll start with the mouth. We'll move through the pharynx and the esophagus into the stomach, then the small intestine, then the large intestine, and we'll talk about defecation. And that is slides number 29 through 100. In the third segment, we'll pick up with the phases of digestion. We've seen this slide before. In the first segment, this was the last slide we looked at, and I used it in order to reinforce mechanical versus chemical digestion, to reinforce organs of the GI tract versus accessory organs. It really constitutes an overview of the digestive system processes that occur in the mouth. This is the beginning of the tube, the fun part, as I like to think about it, right? Where after we take a bite of food, after we take a bite of that cheese sandwich, the tongue and the teeth move that food around our mouth as we masticate, and that food material becomes softened by saliva, this watery solution that includes salivary amylase. So both chemical and mechanical digestion occur in the mouth, and that soft wad of food that you then swallow, that's called a bolus. So that bolus is going to move from the oral cavity into the pharynx, specifically into the oropharynx. There are 3 sets of salivary glands. There are 2 parotid glands, and they're located just anterior to your ear, just deep to the skin. If we sliced open the skin next to your ear, we'd see your parotid gland. And then there are 2 submandibular glands just below the mandible on the right and left side of the mandible. And then there are sublingual glands that are just underneath your tongue. And all 3 of these salivary glands produce the various components of saliva. And saliva is a watery solution. It's a body fluid. And we spoke about salivary amylase as the enzyme that begins chemical digestion of carbohydrates in the mouth. During mastication in the oral cavity, the end result is a bolus, that soft wad of chewed food that is then swallowed. And the process of swallowing is called deglutition. And it's quite a complicated process that involves coordinated action of many skeletal muscles to move that food material into the esophagus, not into the nasal cavity, not into the trachea, but into the esophagus. So there are a couple of safety features in place here to ensure that the bolus ends up in the esophagus. So number 1, and I'll circle it, is this soft palate. So the soft palate, while we swallow, the soft palate rises to block the passageway between the oropharynx and the nasopharynx. So when we swallow, food can't go into our nasal passages. You know it's not a perfect system. Have you ever had food or beverage come out your nose? Many of us have. I hear that wasabi is quite painful, but ordinarily the nasopharynx is there to protect us. The second safety feature involves the epiglottis. The epiglottis is a flap of cartilage that during deglutition, it will descend to cover the trachea. So it blocks the airway, and that helps funnel the bolus into the esophagus, which is exactly where we want it. These protective measures are not perfect. I know this function as snarfing. That's a colloquial term for when food or beverage comes out of your nose. and many of you have had that experience. What are the circumstances? What's going on around you when that happens? We're laughing, right? We're enjoying a meal with friends or family. Somebody says something really funny right as you're in the middle of eating, and that food ends up coming out your nose. And then many of us have experienced choking. So choking occurs when the epiglottis fails to divert the food material away from the trachea, and that food material ends up in the trachea and perhaps even deeper, perhaps in a bronchus or bronchi. And that's called choking, and it's a life-threatening situation, right? Because it can interfere with pulmonary ventilation. And so what's the treatment for choking? The treatment for choking, the initial treatment now is 5 hard slaps on the back. And if that doesn't work, then we perform abdominal thrusts, which were previously known as the Heimlich maneuver, but it turns out Dr. Heimlich wasn't really an ethical scientist. He was quite an unethical scientist. And so we no longer call it the Heimlich maneuver, but rather we perform abdominal thrusts. And during an abdominal thrust, our goal is to push up on the diaphragm in order to increase intrathoracic pressure. We want to raise the pressure inside the thoracic cavity in order to get that food material to move from an area of high pressure to an area of low pressure. And when abdominal thrusts are successful, often there's a popping sound as that food material is dislodged from the airway and it flies out of the person's mouth. So following deglutition, the bolus is in the esophagus, and the esophagus is a passageway between the pharynx and the stomach, and it does not rely on gravity to get your food into your stomach, but rather it relies on a wave-like muscular action called peristalsis. And both layers of the muscularis play a role in peristalsis, and this isn't limited to your esophagus. Peristalsis occurs in your small and large intestine as well. During peristalsis, the circular layer of the muscularis, the deeper layer, that contracts behind the bolus of food, as we see in the diagram that's on the left side. The circular layer contracts behind the bolus, and the longitudinal layer contracts ahead of the bolus, shortening the length of the esophagus. So I think about that shortening as putting on a dress sock or maybe a pair of pantyhose. When you scrunch it up before you put your foot in, that's what's happening in your esophagus, scrunching it up ahead of the bolus or ahead of your foot. And so peristalsis is a wave-like muscular action that propels food material along the GI tract. The stomach is a muscular sac, and I liken the stomach to a physical inbox on somebody's desk. Have you ever had a job where there is a physical inbox on your desk and people put work in it and then you can work on that work later? The purpose of a physical inbox, just like we have an electronic inbox now, like in your email, the purpose of a physical inbox on your desk is because a worker cannot possibly process every bit of work immediately. And so that work is allowed to accumulate and you can process it little bits at a time as you're able. The stomach is like that. You eat a whole meal and the digestive system is just not able to process, to mechanically and chemically digest that food, to absorb that food. It just can't do that all instantaneously. And so that meal or that cheese sandwich, every bite of that cheese sandwich, every bolus lands in your stomach. And so it's your digestive system's inbox. And your stomach, the walls of the stomach produce gastric juice. Gastric means stomach. And gastric juice is a body fluid. It has a low pH. It includes enzymes. We're going to talk about pepsin, which is our first protein digesting enzyme. And because the gastric juice has such a low pH, it's very acidic, like pH 2. the lining of the stomach is protected by a pretty thick layer of mucus so that acid doesn't actually sit on top of those epithelial cells. There's a layer of mucus in between. Here we're looking at the layers of the GI tract, specifically in the stomach. So we talked about the 4 layers of the GI tract in segment 1, and they are the mucosa, the submucosa, the muscularis, and the serosa. So let's talk about things that are specific to the stomach here. In the mucosa of the stomach, there are gastric pits. And these gastric pits are invaginations of the lining, and the cells that line the gastric pits produce the components of gastric juice. Some of them produce mucus, and as we'll see later, there are also enteroendocrine cells here that make gastrin. We're not there yet, but where we are at is gastric juice. So gastric juice is a watery solution that has a very low pH, and it includes a protein digesting enzyme called pepsin. More on that in a bit. The next salient feature of the wall of the stomach is the muscularis. The muscularis of the stomach includes 3 layers of smooth muscle. Everywhere else in the GI tract, the esophagus, the intestines, have 2 layers. There are 3 layers of smooth muscle in the stomach, and they wrap around the stomach at different angles. And so they're able to, when each layer contracts, it's able to compress or constrict the lumen of the stomach in a different dimension. Here's a photomicrograph of the lining of the stomach, where each of these little circular-ish structures here, I'll circle one. I don't know if you can see that. I'll circle one. That's the apical surface of a simple columnar epithelial tissue. And so the stomach mucosa includes simple columnar epithelial tissue, and then these big holes that we see here. These are the openings to the gastric pits, and those pits are lined with cells that are producing the components of gastric juice. We also have cells here that make mucus, because these cells are all, and maybe I can just sort of scribble so that you get a sense, everything here is coated with a thick layer of a thick layer of mucus. And that's because gastric juice has a very low pH, and these cells might be damaged by that low pH. And so the mucus is a protective coating. Gastric juice is a body fluid, and you know that all body fluids are mostly water with a variety of dissolved solutes. And the 2 solutes that we're most interested in here today are an enzyme called pepsin and then hydrochloric acid. So the hydrochloric acid is an acid, and that proton that hydrochloric acid liberates when it goes into solution contributes to the very low pH of gastric juice. Gastric juice has a pH of between 1 and 3. And this low pH takes energy to produce, and it serves several purposes. Number 1, everything that you eat lands in an acid bath. Step 1, acid bath. And that helps to kill any potential pathogens that we have ingested. So whenever we eat, we think that we're so hygienic and we're so clean, but we're really not. With every bite of food, there's gobs of bacteria and fungi and virus particles. And most of them are not going to make you sick, but some of them have the potential to make you sick. So in order to denature their proteins, right, all living things can include proteins, viruses include proteins, as a protective mechanism, every bite of food that you eat lands in gastric juice. It lands in an acid bath. The second function of this low pH of gastric juice is that it causes proteins to denature. I hope you remember that one of the chemical factors, one of the environmental factors that influences protein folding is the local pH. And that local pH is going to influence these inter and intramolecular forces that help a protein to fold, only at this very low pH, it will help a protein to unfold, and that's called denaturing. And a denatured protein is more accessible. The amino acid, the peptide bonds between amino acids become more available to protein digesting enzymes as this, think about untying or unraveling a knot. So now the whole string is available to be cut up by enzymes, and these enzymes are little molecular scissors. So the low pH of gastric juice kills ingested pathogens, it denatures proteins, and we require this thick layer of mucus lining our stomach in order to protect ourselves from the low pH of gastric juice, as well as from the pepsin, this protein digesting enzyme that's in the lumen of the stomach. So pepsin is a protease, and a protease is a protein digesting enzyme. And I don't like the name pepsin because it doesn't end in ase, but it's an enzyme. So sometimes it throws students off. So pepsin is an enzyme, even though the name doesn't end in ase. And pepsin catabolizes proteins into smaller peptides, and eventually those peptides are going to be catabolized into individual amino acids. Although just like in the mouth with carbohydrates, carbohydrate digestion begins in the mouth, it's not finished there. That also applies in the stomach with proteins. So in the stomach, the digestion of protein begins, but it's certainly far from complete. Proteins will be further digested in the small intestine. So pepsin is an enzyme that requires a low pH in order to function. At a higher pH, pepsin changes shape. Enzymes are proteins. Proteins are influenced by the local pH. So pepsin is in its optimal shape in the low pH of gastric juice. When that pepsin moves into the small intestine, along with the liquidy food material that moves into the small intestine, we'll see that the pH comes up. And so in the more neutral to alkaline environment of the small intestine, pepsin stops working. So pepsin is active in the stomach and it's active in the small intestine until it's sufficiently inactivated by the higher pH in the small intestine. So here we're looking at the wall of the stomach, gastric pits, and the different cell types that line gastric pits. So you're not responsible for knowing the names of the different cell types or their individual functions, but some of these cells are responsible for manufacturing pepsinogen. And there's that ogen again. We've encountered this before in the cardiovascular system when we looked at the clotting cascade. Ogen is a suffix that indicates that the molecule is an inactive precursor. In the clotting system, we talked about fibrinogen, for example. Fibrinogen is inactive. It's a precursor to fibrin. Well, that applies here. Pepsinogen is inactive. It's a precursor to pepsin. Pepsin is the active enzyme. And a couple of places in the digestive system, protein digesting enzymes are secreted as zymogens. Actually, protein digesting enzymes in the digestive system are always initially secreted as zymogens. And a zymogen is an inactive precursor of an enzyme. So here in the stomach, these cells secrete pepsinogen. Pepsinogen doesn't do anything. It's harmless. And that's the point. So cells are full of proteins. There are proteins everywhere in cells. And now imagine you're a cell. You're made out of protein, and of course all the other biological molecules, but you're made largely out of protein, and it's your job to manufacture a protein digesting enzyme. That's dangerous stuff. If that protein digesting enzyme leaked out or got loose, it would start destroying proteins everywhere. And so the way around this is for the cell to produce an inactive, inert, harmless precursor. So these cells secrete, they spit out essentially pepsinogen, and that's fine. And then pepsinogen gets converted into pepsin in the lumen of the stomach. And it's shown here on this diagram. When that pepsinogen encounters either hydrochloric acid or molecules of pepsin, that pepsinogen, part of that molecule gets cut off, and pepsinogen is converted into pepsin. And now pepsin is the active protein digesting enzyme. And now if you think about the cells that line the gastric pit, now there's this protective layer of mucus between the active, dangerous pepsin and the cells themselves. Here's another artistic representation of the same process. So we're looking at a gastric pit, and we see that one type of cell, the chief cells here, secrete pepsinogen. That's an inert, inactive, harmless substance. And then once that pepsinogen arrives in the lumen of the stomach, it gets activated, and it's activated by either hydrochloric acid or existing molecules of pepsin. And that pepsinogen is activated into pepsin, and then pepsin catalyzes the catabolism of protein into smaller peptides. So this is our beginning of protein digestion, the chemical digestion of protein in the stomach. When the food material is in the stomach, two sphincters at either end of the stomach seal off the stomach. So the lower esophageal sphincter is a ring of smooth muscle that occurs at the junction of the esophagus and the stomach. And then the pyloric sphincter is also a ring, a circular smooth muscle, and it occurs at the junction between the stomach and the first region of the small intestine. And so after you've eaten a meal, you finish your cheese sandwich, for example, both of these sphincters contract, and the walls of the stomach from those gastric pits exude or secreted many milliliters of gastric juice. And so this sort of becomes like a sealed off Ziploc bag. And then the walls of the stomach, those three layers of smooth muscle, begin to contract. And this is called gastric churning. So during gastric churning, all of those boluses that you swallowed, those wads of food that you swallowed, they get macerated by the gastric churning, and they get moistened by the gastric juice, and ultimately your meal is liquefied. And that liquid that's produced in your stomach is called chyme, C-H-Y-M-E. If that lower esophageal sphincter is leaky, if it doesn't create a tight seal, you might experience symptoms of gastric reflux, also known as heartburn, but heartburn is a misnomer because it's really not about your heart. It's acid, gastric juice, and food material leaking out of your stomach and up into your esophagus. The mucosa of your esophagus, that epithelial tissue in your esophagus, is not protected by a thick layer of mucus. And so even though it has a protective layer of epithelium, that epithelium is kind of vulnerable to the low pH, and it can cause damage to that tissue. It can cause an ulceration, and you might experience that as pain, known as heartburn. And most of us have experienced heartburn, or we experience it occasionally. Some people deal with chronic heartburn. They have heartburn regularly. And that's a problem because regular heartburn is known to increase the risk of esophageal cancer. So it induces changes in those esophageal cells, and those cells can become cancerous. So for the person who experiences regular or chronic heartburn, they want to do whatever they can to keep that under control. And some ways to prevent or control heartburn is to avoid acidic foods, which will further lower the pH of gastric juice. Avoid caffeine. Caffeine has lots of effects in the body. We recently talked about how caffeine acts as a diuretic, especially in higher doses. But caffeine can also promote the production of hydrochloric acid in the stomach. And that's the problem. And we want to avoid the problem, so avoid caffeine. And then in order to avoid gastric reflux, don't eat right before you go to bed. So think about the body in the vertical versus the horizontal position and what makes gastric reflux easier. Standing up, can gastric reflux occur? Absolutely. Is it easier when the body is laying down? Is it easier for those fluids to leak back up through your leaky sphincter into your esophagus? Yes. So avoid eating for 3 hours before going to bed. And over those 3 hours, that meal will slowly make its way out of your stomach and into your small intestine. So the risk of gastric reflux decreases over time following your meal. And then treatments include antacids. These are over-the-counter medications that can raise the pH of gastric juice. So maybe you drink sort of like this chalky fluid like Mylanta. That's going to help raise the pH of gastric juice so it becomes less dangerous to your esophagus. And then there are also over-the-counter drugs that slow the production of hydrochloric acid, slow the production of that acidic gastric juice. So these are they used to be prescription, but now they're available over the counter. These are two different medications that you can take to prevent heartburn. And they reduce the secretion of hydrochloric acid. And there are two different categories of drugs. I just selected two examples here. So Prilosec is an example of a proton pump inhibitor. And so we're not studying how cells secrete hydrochloric acid. However, these proton pump inhibitors are able to bind to and disrupt the function of a proton potassium ATPase. So there are these pumps in the membrane, and if we can slow down these pumps, we slow down the rate at which these cells are able to secrete hydrogen. And so we slow down, or we raise, the pH of gastric juice. And then the other drug class shown here are histamine blockers, H2 blockers. And so histamines are signaling molecules in the body, and histamines can cause the production of hydrochloric acid. And so if we can take an antagonist, a histamine antagonist, remember an antagonist is a competing signaling molecule that will bind to and block the action of that receptor. So we can block the effects of histamine and reduce the pH of gastric juice. Increase the pH of gastric juice. Reduce the secretion of hydrochloric acid is a better way to say that. So throughout the GI tract, we'll be talking about motility, and we have been talking about motility. Motility refers to the mixing and propulsion. How are the contents of the GI tract churned around, stirred up, and how are they propelled toward the anus? So in the stomach, the type of churning that happens, the mixing, is called gastric churning. And so during gastric churning, both of our sphincters are closed off. The lower esophageal sphincter is sealed, the pyloric sphincter is sealed, and then gastric churning occurs. Those 3 layers of smooth muscle contract in different planes, and that food is liquefied. It's turned into a liquid called chyme, C-H-Y-M-E. Don't say chime, you'll sound really silly. And so now this cheese sandwich that we ate, and it was solid, now it's liquid as it gets mixed with gastric juice, and it's called chyme. And after some time, it depends on how long, depends on what you ate, and we'll talk about that later. But after an hour, 2, 3 hours, the process of gastric emptying begins. And during gastric emptying, the pyloric sphincter starts to open. It opens just a little bit. There's a crack in the door. And with every mixing wave, with every movement of the muscularis of the stomach, a little bit of the chyme is forced through the pyloric sphincter into the first region of the small intestine, which is called the duodenum. And so this can take hours for gastric emptying to be complete. A few milliliters of chyme move at a time from the stomach into the small intestine. So I mentioned this earlier, I think in segment 1, that the contents of the GI tract are not considered part of your body, but rather they're considered to be external to the body. So your GI tract can be seen as a tube through which the external environment passes through. So you're sort of like a big donut. And it is the process of absorption, which most absorption happens in the small intestine, but that process of absorption is what brings those molecules and ions from the external to the internal as those things are added to your blood. So we will talk about absorption in the stomach. A little bit of absorption happens in your stomach, but not much. So most absorption happens in your small intestine. What can be absorbed in your stomach is shown here. Water, which always moves by osmosis. Water can be absorbed in your stomach. Alcohol, which is a small molecule, it's pretty lipid soluble. So alcohol can cross that mucosa by diffusion. And then a few other drugs, and they're all small and fat soluble. Maybe you've had the alcohol experience. If you've ever had an alcoholic beverage on an empty stomach, you feel the effects very quickly. But after a meal, let's say you're drinking a glass of wine with a meal, most of that alcohol will be absorbed with other molecules in your meal when we get to the small intestine. So moving into the small intestine, we're going to talk about chemical digestion. We're going to talk about absorption in the small intestine. A lot happens in the small intestine. It's the busiest place in your digestive system. So most chemical digestion and most absorption occurs in your small intestine. And your small intestine gets help from accessory organs, the pancreas, the liver, and the gallbladder. I love this diagram. It's pretty simple. It shows the plumbing relationship, the structural relationships between the liver, the gallbladder, the stomach, the small intestine, and the pancreas. The arrows show the direction of movement of chyme, bile, pancreatic juice, and intestinal juice. So we talked about gastric emptying, and during gastric emptying, it occurs over time. Small bits of chyme are moved from the stomach into the duodenum. The duodenum is the first region of the small intestine, followed by the jejunum and the ileum. Duodenum, jejunum, ileum. Those are the 3 regions of the small intestine. Here we are in the duodenum, the busiest part of the small intestine. So the duodenum receives acidic chyme as that chyme passes through the pyloric sphincter. The wall of the duodenum includes glands that produce intestinal juice. This intestinal juice has an alkaline pH to help raise the pH of the chyme. We don't need to be so acidic anymore. We've been through our acid bath, and acid requires extra protective measures. So let's not be acidic anymore. So intestinal juice has a relatively high pH. Pancreatic juice is produced by cells in the pancreas, and that pancreatic juice is delivered into a series of ducts that then connect into the duodenum. And then we have bile. Bile is manufactured by liver cells, and bile can be stored in the gallbladder, And bile can be delivered both from the liver as well as the gallbladder into the duodenum. So the duodenum is this rich mixture. We have all of the food molecules in that cheese sandwich. We have gastric juice, there's mucus, and then to that we add intestinal juice, pancreatic juice, and bile. So the chyme that's in the small intestine is a very rich mixture of molecules from very different sources. The liver performs many, many different functions, and it contributes to your overall homeostasis numerous ways. We're in the digestive system now, and so obviously we're going to focus on the liver's contribution to the digestive system. And that contribution is to manufacture bile and to deliver that bile into the duodenum. So we've talked about bile before. When we talked about hemoglobin recycling, we talked about this waste product called bilirubin, and bilirubin is released into your blood and it gets taken up by your liver cells. That bilirubin gets incorporated into bile, and bile has a yellowish green appearance. And the gallbladder, which is a storage sac for bile, has this dark green appearance. If you've ever seen a cadaver, maybe you've dissected some vertebrate, you have seen a gallbladder on the inferior aspect of the liver before. So bile is this yellowish green pigment that's produced by liver cells and stored in the gallbladder. And as we'll see, bile acts as an emulsifier. So bile does not contain any digestive enzymes, but it helps keep fat droplets small in the chyme. So I think this is the first time we're encountering the word emulsification or emulsifier. So to emulsify means to break up lipid droplets and to keep them broken up, to keep them as small lipid droplets in a watery medium. So think about a very simple salad dressing in a jar on your counter, and it consists of olive oil and vinegar. Vinegar is mostly water. Olive oil is mostly triglyceride. So we have this mixture of oil and water, and you know that they don't mix, and you know that the oil is sitting on top of the vinegar. In order to emulsify that mixture, you'll pick up the jar and you'll shake it vigorously. So you'll physically break up that big fat droplet into many small fat droplets. And for a few moments, those little fat droplets are surrounded by the water in the vinegar, and you pour that onto your salad. And then you put the jar back down on the counter, and what happens over the next few minutes? It separates again. All of those little oil droplets find each other and merge into bigger and bigger oil droplets until we have total separation. All of the oil is on the top and all of the water or the vinegar is on the bottom. Well, we don't want that separation to happen in the chyme that's in your small intestine. We need those fat droplets to not find each other and merge into bigger fat droplets. We want those little fat droplets to stay little. Those little fat droplets will have more surface area in order for a lipid digesting enzyme to break down to chemically digest those triglycerides. So bile acts as an emulsifier in the small intestine, and what that means is bile prevents these small fat droplets from aggregating, from combining to form larger fat droplets. And this is going to make for more effective chemical digestion of these lipids by a particular enzyme called pancreatic lipase. So that's a lipase, a lipid digesting enzyme that was produced by the pancreas. And the way that bile can act as an emulsifier, how it accomplishes this task, is because bile, these molecules sometimes referred to as bile salts, they are amphipathic. Here's one way to visualize emulsification. So in that cheese sandwich, cheese, lot of lipid in cheese, a lot of fat in cheese. And you remember, fat and water don't mix. And so in that chyme, we might have a large lipid droplet surrounded by water. And we want to break up that lipid droplet into smaller lipid droplets and keep those smaller lipid droplets separated from each other to prevent them from aggregating together to reform this large lipid droplet. So these are the bile salts, and they have a familiar shape to you. Maybe you recognize that there's a polar head and non-polar tails, and so they're amphipathic. And what happens when we add bile to chyme that has large lipid droplets in it, and with that gastric churning will essentially shake up your salad dressing. It'll break up the large lipid droplets into smaller lipid droplets, and then these smaller lipid droplets are surrounded by bile salts. And so these cages of bile salts prevent these lipid droplets from smashing into each other and forming a larger lipid droplet. So this is the process of emulsification. Here's another artistic representation of the process of emulsification, as well as its importance. So without an emulsifier, these small fat droplets will merge together to form this very large fat droplet. And then we have these sort of angry looking and sad looking molecules of lipase. Lipase is a triglyceride digesting enzyme. And so when we have this very large lipid droplet, there's not a lot of surface area for the lipases to work. And so these two lipases are sad because there's no room for them. They can't reach the lipid droplet. And so lipid chemical digestion will be limited because there's just not enough surface area for all the lipase to get a piece of the action, for all the lipase to be active or to be exposed to the lipid. So in the panel below, now we have lipid droplets that are emulsified. They are surrounded by bile. And so now we have a much greater surface area. We have a much greater lipid surface area so that every single lipase molecule available has access to lipid. Alton Brown is a celebrity chef, television food personality, and many years ago, for many years, he made a darling low-budget food program called Good Eats. And he's very scientifically minded, and he takes opportunities to explain what's happening with the chemistry and the physics and the biology of the food as you prepare it. And we're going to watch the first 9 minutes or so of an old episode of Good Eats called Mayo Clinic. And it's about making mayonnaise. And Alton's purposes are to teach you about making mayonnaise. Mayonnaise is an emulsion. There's an emulsifier involved. And my purposes are to teach you about emulsification. So we'll watch about the first 9 minutes of this video. Okay, I hope Alton has contributed to your understanding of emulsification. Alton also has a new series called Good Eats Reloaded, where present-day Alton goes back and examines some of his thinking and some of his recipes and makes corrections. And that's what a good scientist does. It goes back and re-examines the evidence, and a good scientist goes back and corrects his or her thinking. So we are in the small intestine. We are in the duodenum, the first region of the small intestine. And that's a super busy place in your GI tract. Remember, the duodenum receives chyme from the stomach. It receives bile from the liver and the gallbladder, and it receives pancreatic juice from the pancreas. The wall of the duodenum itself also makes intestinal juice. So there's a very rich mixture of partially digested molecules. Here's the role of the pancreas in the digestive system. So the pancreas is both an endocrine organ and a digestive system organ. As part of the digestive system, cells in the pancreas produce the components of pancreatic juice, and pancreatic juice has an alkaline pH, and it includes many different enzymes. There are carbohydrate digesting enzymes. There are lipid digesting enzymes. There are nucleic acid digesting enzymes, and there are protein digesting enzymes that are part of pancreatic juice. In this diagram, we can see the plumbing relationship between the pancreas and the duodenum, the first region of the small intestine. And there are many different types of cells in the pancreas. They can be divided into two categories. There are acini cells, or sometimes they're called acinar cells, and those cells make up the bulk of the tissue of the pancreas. And the acinar cells secrete the components of pancreatic juice, and they secrete those components directly into ducts. In this image, the pancreatic ducts are in green, as well as the bile duct, actually. Ducts are in green. And a duct in this image is a passageway for a secretion to flow into the duodenum. The second type of cell or category, group of cells in the pancreas make up what are known as pancreatic islets, or they used to be called islets of Langerhans. And the islet cells secrete hormones, either insulin or glucagon, and they are part of the endocrine system. They're not considered part of the digestive system. So these acinar cells secrete the components of pancreatic juice, and pancreatic juice is secreted into the duodenum. So we have a different representation of the pancreas and the duodenum here. Also included on this image is the gallbladder and the bile ducts. So you can see that bile and pancreatic juice enter the duodenum at the same point. And pancreatic juice has an alkaline pH, as does bile. Bile also has an alkaline pH. And this higher pH helps to neutralize the acids that are in chyme. So as this acidic chyme moves into the duodenum from the stomach, the pH slowly rises as that chyme becomes mixed with bile, pancreatic juice, as well as intestinal juice. And that higher pH is going to inactivate the pepsin that was active at the low pH in the stomach. Pepsin is going to gradually become inactivated as it changes shape slightly due to the higher pH. There are many different digestive enzymes in pancreatic juice. They digest carbs, they digest lipids, they digest proteins, and there are also nucleic acid digesting enzymes that are part of pancreatic juice. So these are the example enzymes that we're going to cover. We're not covering all of the pancreatic enzymes, rather a select few. So an example of a lipid digesting pancreatic enzyme is pancreatic lipase. A carb digesting enzyme is pancreatic amylase. And a protein, we'll do two protein digesting enzymes, trypsin and chymotrypsin. And again, just like we saw with pepsin, pepsin's also a protein digesting enzyme. Pepsin works in the stomach. These two protein digesting enzymes from the pancreas, trypsin and chymotrypsin, they are initially secreted as zymogens. A zymogen is an inactive protein digesting enzyme. And these zymogens are then activated in the lumen of the small intestine. And again, this is a protective feature. When a cell manufactures protein digesting enzymes, that cell is very vulnerable to those protein digesting enzymes. So for that, the safety of that cell, the protein digesting enzymes are produced and secreted as zymogens. Zymogens are inactive, so zymogens are not dangerous to the producing cell, and they are only activated once they're in the lumen of the small intestine. Pancreatic juice includes the zymogens trypsinogen and chymotrypsinogen, and there's that suffix again, ogen. Ogen is a clue that it might be an inactive precursor to something, and that's true in this case. So these two substances, these two zymogens, are part of pancreatic juice, and they are cleaved into their active form by a particular enzyme that's present in the small intestine, and that enzyme is called enterokinase. And enterokinase will be our introduction to brush border enzymes. Enterokinase is a brush border enzyme. We will cover just two brush border enzymes, enterokinase, and a little bit later we'll talk about lactase. But there are many different brush border enzymes, and they are an important component of chemical digestion. Here we're looking at what's called an absorptive cell that is part of the mucosa or the lining of the small intestine. And these absorptive cells, importantly, they have microvilli. Microvilli are sometimes described as finger-like projections of the plasma membrane. So this is the apical cell surface. It's lining the lumen, and that's where the food material is. And so these microvilli are a way of augmenting, of amplifying the amount of surface area present on that cellular surface. And embedded in the plasma membrane of these microvilli are brush border enzymes. There are many different types of brush border enzymes. We're going to cover two, enterokinase and lactase. Right now we're thinking about enterokinase. This is what the brush border looks like through a light microscope. So those are simple columnar epithelial cells, and they have microvilli, and collectively they create this fuzzy looking border, which is why it's called the brush border. Embedded in that brush border are many different enzymes. They're part of the plasma membrane here. So trypsinogen is the first zymogen to be activated, and it's activated by enterokinase. Enterokinase is an enzyme, and it cuts off a particular portion of the trypsinogen molecule, yielding trypsin. Trypsin is now an active protease. It can chemically digest proteins. Trypsin can also activate chymotrypsinogen. So in the presence of trypsin, chymotrypsinogen is activated and it becomes chymotrypsin. And so both trypsin and chymotrypsin are active protein digesting enzymes. And so here we are in the lumen of the small intestine. We see trypsinogen and chymotrypsinogen. They are secreted in their zymogen form. This image also includes another zymogen that we're not covering, procarboxypeptidase. And we can see that trypsin is the first enzyme to become activated. And it's activated by the brush border enzyme enterokinase. And once we have trypsin, trypsin can then go on and activate other zymogens. So it'll take chymotrypsinogen and convert it into chymotrypsin. So in this very busy place called the small intestine, we have a very rich mixture of partially digested food molecules. We have dozens of different enzymes that are present in the brush border, that are present in pancreatic juice, and we have bile as well as intestinal juice. And so the greatest amount of chemical digestion, the greatest amount of enzymatic activity occurs in the small intestine because we have the greatest number of enzymes present. So here we can take a look at the chemical makeup of various carbohydrates that we find in food, and they're listed by size, right, from largest to smallest. Polysaccharides include starch, cellulose, and glycogen, and the monomer building block of all of those, starch, cellulose, and glycogen, is glucose. Disaccharides, you recall, are made up of two monomer building blocks, two monosaccharides linked together. And the most common disaccharides we find in food are sucrose, that's table sugar, lactose, that's milk sugar, and maltose. And you can see there are different combinations of monosaccharides, like sucrose is one glucose attached to one fructose. And then it's the monosaccharides that we actually absorb. So we eat these carbohydrates in food, and if we want to make use of these carbohydrates, they need to be absorbed. and the only form in which they can be absorbed is in their monomer building block or monosaccharide form. So we can absorb glucose, fructose, and galactose. We cannot absorb disaccharides like lactose. We cannot absorb lactose. It needs to be cleaved. And so that last stage of processing of chemical digestion for the carbohydrates often occurs via a brush border enzyme. So there are 3 different brush border enzymes shown here: maltase, lactase, and sucrase. And they each have their unique substrate. Maltose is the substrate for maltase. Lactose is the substrate for lactase, and so on. And only following the action of these brush border enzymes do we get the carbohydrates into their monosaccharide form. And now these carbohydrates are small enough to be absorbed. So as adults, not everybody produces lactase, and it's one way that our species differs genetically around the world. So if you're lactose intolerant, it means that in your brush border, we don't find very much lactase. And it suggests that somewhere in your deep, deep ancestry, in your deep evolutionary history, so we're not even talking about your great-great-grandparents, we're going back tens of thousands of years. Somewhere in your genetic evolutionary history, if you are lactose intolerant, part of your genome comes from, or maybe all of your genome comes from a region of the world where dairy cultivation was not a practice. And if you are lactose tolerant, or it's called lactase persistent, if you are able to consume dairy products with no digestive system issues, then it suggests that you continue to make lactase. If we look at your brush border, we'll find that enzyme, and that enzyme allows you to digest lactose and to absorb the resulting monosaccharides. So for people who are lactose intolerant, meaning they don't make enough lactase, what happens when they drink cow's milk or when they eat ice cream? They ingest a dairy product. The symptoms of lactose intolerance include flatulence, and that's the fancy word for passing gas or farting. There can be intestinal cramping, and there can be diarrhea. What causes this? Well, if there's lactose, milk sugar, in the small intestine, but there's no lactase to break it down, that lactose, the disaccharide, cannot be absorbed, and it continues to move along the lumen of the small intestine until it arrives in the large intestine. And in the large intestine, we have a rich mixture of bacteria, and they're part of our microbiome. They belong there in the large intestine. We have a mutualistic relationship with these bacteria. They keep us healthy. They make certain vitamins for us, and we feed them. It's an exceptional circumstance when a lactose intolerant person ingests this milk sugar, because it ends up in the large intestine. The lactose did not get digested or absorbed in the small intestine, and so the bacteria get this pretty big influx of sugar, a disaccharide, that the bacteria can make use of. So they get this big influx of calories, this big influx of nutrition. And so bacteria do what bacteria do. They eat and divide and eat and divide. And this rapid proliferation of bacteria can irritate the lining of the GI tract. Bacteria produce gases, just like your cells produce gas. So that can lead to the flatulence, and the irritation can lead to cramping and diarrhea. The body responds with, get it out. For most of human evolutionary history, the default trait is lactose intolerant. The only human to drink milk is a baby human. And so our ability to produce lactase into adulthood was never selected for. And so as far as we can tell, everyone for most of human evolutionary history was lactose intolerant, just like many people are today. But this new trait called lactase persistent, which confers the ability to derive nutrition. You can digest that lactose and you can get those calories. That evolved in our species twice. And we know from a combination of archaeology, anthropology, and population genetics, we know that it evolved twice, and it evolved independently as two different peoples in two different places in the world began the practice of dairy agriculture. So raising cows and goats, and then not only using them for meat, but using them as a source of milk. And remember, the default mode is to be lactose intolerant. So if you're in one of these populations and there's a severe drought and famine sets in, the crops fail, but we still have these cows and goats and they still produce milk. If you're the individual in that population and you have, just by chance, you have that random mutation that allows you to process lactose to derive nutrition from it, well, then you're going to survive the famine better than your peers who might all be lactose intolerant. And so you're going to have more offspring than your peers, and those offspring are also going to carry that lactase persistent trait. And so this trait in a dairy cultivating culture, that trait will spread very quickly through the population. That happened twice. It happened here in Northern Europe, and it happened here in Western Africa. Of course, people spread around the planet. A lot of European colonization happened in North America and Australia and South America, and they brought their lactase persistent genes with them. But it didn't happen everywhere. So if your deep ancestry comes from part of the world where dairy cultivation was not really a practice, then chances are you are lactose intolerant. This is a fantastic video about lactose intolerance and the evolution of a novel trait called lactase persistence. And it's made by HHMI, which is Howard Hughes Medical Institute, and it's well worth 15 minutes of your time. So go and Google this video, spend 15 minutes learning about human physiology from the global evolutionary perspective, and then rejoin us here as we continue our journey through the small intestine. So your small intestine is where the greatest amount of chemical digestion occurs. It's also where the greatest amount of absorption occurs, and we'll talk about absorption soon. So the lining of the small intestine is critical for both digestion because of those brush border enzymes, as well as absorption. In order for a material to be absorbed, it has to cross the lining of the small intestine. And the more lining you have, the more opportunity there is for absorption. And so the small intestine has a whole bunch of anatomical features to increase the surface area of the lining of the small intestine. So first of all, your small intestine is very long, right? The longer the small intestine, the greater the opportunity for digestion and absorption. So your small intestine is about 20 feet long. And then internally, the lining has these big folds in it, and those are called circular folds. I'll show you a photo in just a moment. And these circular folds increase surface area. They also encourage the chyme to move through the small intestine in sort of a swirling fashion rather than a straight shot. And if the liquid, the chyme, swirls through a tube, it will spend more time in contact with the lining of the tube. And then these circular folds are coated with these finger-like projections called villi, which further increases the surface area of the small intestine. And then of course we have these microvilli. So every cell has a fuzzy border, right, made up of microvilli. So the surface area of just your small intestine is bigger than the surface area of a tennis court. In this diagram, we can see many of those anatomical features. So if we slice open a small intestine, with your bare eye, you can see circular folds. And it's sort of these ridges that we see lining the small intestine. And if we zoom in to just a singular circular fold, we see that the surface of a circular fold is also convoluted and bumpy, and it's covered with these structures known as villi. So here's one intestinal villus being highlighted here. Villus is singular, villi is plural. And so when that one villus is blown up here, we can see these are a mixture of absorptive cells and goblet cells. So the goblet cells make mucus, and here mucus is acting like a lubricant to help keep the chyme flowing smoothly. And these absorptive cells, shown here, here's an absorptive cell with its brush border. It has microvilli on the apical cell surface. And so collectively, when we stack these absorptive cells together, we get that brush border. And so the circular folds, the villi, and the microvilli all serve to augment the surface area in the small intestine. And that maximizes both chemical digestion as well as absorption. As we move through the GI tract from mouth to anus, we encounter different types of motility. And motility just means, well, what is the muscularis of the GI tract? What's the muscularis doing in any particular region in order to mix the contents of the GI tract and to propel the contents of the GI tract downstream or toward the rectum? And so, to remind you, in the mouth and the pharynx, we encountered deglutition. That just means swallowing. In the esophagus, that's the first place we encountered peristalsis. And in the stomach, we encountered mixing waves and gastric emptying. And here we are in the small intestine. And the two types of motility that we see in the small intestine are called segmentations and migrating myoelectric complex. So segmentations can be viewed as a local sort of squishing back and forth. And as that chyme is propelled both forward and backward over a few centimeters, that chyme becomes mixed with the enzymes that are present in the small intestine. So from pancreatic juice, it becomes mixed with chyme, with bile. And that chyme gets more exposure to the brush border enzymes that are part of the mucosa, part of the lining. And then the other type of motility that we observe in the small intestine, migrating myoelectric complex, or some places you might see it written migrating motility complex, same thing, MMC. MMC is this really slow peristaltic wave that actually starts in the distal region of the stomach, and then very slowly, this big wave of muscle contraction migrates very slowly from the stomach all the way to the end of the small intestine, to the ileocecal sphincter, which we'll take a look at in just a little bit. And that takes 2 hours. And so movement through the small intestine is pretty slow. The more time food material spends in the small intestine, the more opportunity there is for chemical digestion as well as absorption. So in this image, we can see the effects of segmentation, this local squishing back and forth. And you can see how different regions of the chyme are mixed together. They're also being mixed with the great variety of enzymes that we find in the small intestine. Most absorption occurs in the small intestine, and I hope you remember that absorption means nutrient molecules leave the lumen of the GI tract by crossing the wall and getting into either a lymphatic vessel or a blood vessel. And we'll take a look at that in just a moment. So the vast majority of digestion occurs in your small intestine. Very little and only certain things can be absorbed in your stomach. Everything else is absorbed in your small intestine. And let's take a look at that. In the panel on the left is an intestinal villus. Villus is singular, villi is plural. So we're looking at just a single villus here. And I'll remind you that a villus is sometimes described as a finger-like projection of the mucosa of the small intestine, the lining of the small intestine. So if we take one of those ridges in the lining, one circular fold, that circular fold is just coated in villi, almost like shag carpeting. And so now we're looking at one villus. And understanding the anatomy of a villus is important for understanding the physiology of absorption. So the vast majority of cells here forming the covering of this villus, the vast majority of them are absorptive cells, and that's the type of cell shown here in panel B. These absorptive cells are they have microvilli on their apical surface, and that gives them more surface area, more contact with the food material. And then moving back over to panel A, the yellow cells are goblet cells here. Goblet cells are labeled here. Goblet cells produce mucus, and in this context, mucus is acting as a lubricant. If we look at the core of the villus, we see two different sets of tubes. In pink and blue, we have blood capillaries, and that's just a regular capillary bed. And so as oxygen-rich blood enters these capillaries, it's going to lose oxygen, so oxygen unloading is going to occur, and carbon dioxide loading is going to occur, and nutrient loading is also going to occur. So we have gas exchange, but we also have blood gaining nutrients, and then these nutrients will be distributed all over the body. The second type of vessel that we have here inside of villus is a lacteal, and here it's shown in green. And what a lacteal is it is a blind-ended lymphatic capillary. So we talked about the lymphatic system when we covered the cardiovascular system, and I hope you remember that the lymphatic system is responsible for collecting excess interstitial fluid and moving that interstitial fluid, which is now called lymph, moving that lymph and eventually returning that fluid to the cardiovascular system. Another function of the lymphatic system is to absorb fat-soluble nutrients, which we'll talk about, and that happens in this lacteal here. So as food material crosses the absorptive cells, that food material is either going to enter a blood capillary or it's going to enter a lymphatic capillary, depending on the type of material. Here we can see what happens to water-soluble nutrients. Monosaccharides is the example here. So we see chemical digestion of the polysaccharide into a disaccharide into two monosaccharides. And then those monosaccharides are not small enough to be absorbed. And monosaccharides, as well as amino acids, they're water soluble. And so these substances cross the absorptive cell and they move right into a blood capillary. So this is why if you eat food that's rich in carbohydrates, this is going to cause your blood glucose to rise. During absorption, that glucose moves into your blood. And fructose, by the way, is easily converted into glucose inside this epithelial cell. Amino acids are also mostly water soluble. So as this protein is chemically digested into peptides and down to individual amino acids, those amino acids follow a similar route. They pass through the absorptive cell and right into the blood. And then those amino acids are now available to all of your body cells. And your body cells will use those amino acids to build proteins or maybe to produce ATP. The way that fats and fat-soluble nutrients get absorbed is a bit more complex. So let's start by examining what's happening in the lumen of the small intestine here. So we have our emulsified fat droplets, and we see that they're emulsified because they're coated in bile salts. These tiny droplets of fat are coated in bile salts. The bile salts are pink here. And we know that in the small intestine we have lipases present. A lipase is a lipid digesting enzyme. And the lipid, the type of lipid that we're dealing with here, is the triglyceride. So those are fats and oils. And triglycerides, when they're acted upon by lipase, triglycerides get digested into fatty acids and monoglycerides. A monoglyceride is a glycerol with only one fatty acid attached. So triglycerides have 3, a monoglyceride has 1. The other 2 fatty acid tails were liberated or broken off by lipase. So following the activity of lipase, we have these micelles, these tiny little spheres of digested lipids coated in bile salt. And the fatty acids and the monoglycerides, they're now small enough and fat soluble enough to zoop right across the phospholipid bilayer of this absorptive cell. And so every time a mycel bumps into the surface of an absorptive cell, those monoglycerides and fatty acids leave the mycel, leave some of the bile salts behind in the small intestine, and those fats move into the absorptive cell. Once inside the absorptive cell, the story gets further complicated, and I'll use the next diagram to explain that. But before we leave this diagram, I want you to notice, please, that these newly absorbed fats don't enter into a blood capillary. It turns out that they're too big. They can't cross the wall of the capillary, but they can get into a lacteal, and a lacteal is the lymphatic vessel. So in this diagram, we can compare the absorption of carbohydrates and amino acids with the absorption of fat-soluble things. So we talked about the absorption of carbohydrates and amino acids. They're water-soluble, and they cross the plasma membrane of the absorptive cell by a variety of transport mechanisms that you recognize. You recognize this. And one way or another, they pass through the absorptive cell, and they're all water soluble enough, and they go right into the blood. And now this is nutrient loading as that blood passes through your small intestine, and you've eaten a meal recently. We can continue the story of what happens with triglycerides down here. So we'll start in the lumen of the small intestine, and we have these mycels. A mycel is a sphere of digested lipids, and it's coated in bile salts. The bile salts are not really well represented here. Maybe it's that black line that forms the circle. And so inside that mycel, we find fatty acids and monoglycerides. Remember, these used to be triglycerides, but they were chemically digested by lipase. And every time that mycel bumps into this apical cell surface, these small fat-soluble things, zoop, go through simple diffusion as they cross that plasma membrane. Now, this is where the next layer of complexity occurs. Inside the absorptive cell, these digested lipid components recombine into triglycerides, which seems really inefficient. Why go to the trouble of chemically digesting them if they're only going to reform inside these absorptive cells? Well, there's no other way to get them into the cell. So we have to chemically digest them into building blocks, monoglycerides and fatty acids, in order to get those materials into the cell. Then once they're inside the absorptive cell, they reform into triglycerides, and that triglyceride droplet gets coated in a layer of phospholipids to form this pretty large structure called a chylomicron. A chylomicron is one type of lipoprotein. It's a giant lipoprotein. I'll show you an image of a lipoprotein in just a little bit. And these chylomicrons leave the absorptive cell by exocytosis because they're so big. That's the only way that they can get across the plasma membrane. And these chylomicrons are too large to cross the wall of a capillary. They don't fit through those intercellular clefts in the capillary wall, but they do fit in the spaces in the wall of a lymphatic capillary, the wall of a lacteal. So these chylomicrons move into the lacteal that's part of an intestinal villus. Here's a third artistic representation of the same process. It's a little bit complicated. So here we have our mycels in the lumen of the small intestine. And so we have digested triglycerides here, and that means we have monoglycerides and fatty acids coated with bile salts. Every time one of these mycels bumps into the surface of an absorptive cell, those monoglycerides and fatty acids diffuse across the plasma membrane and enter the absorptive cell. Inside the absorptive cell, we reform triglycerides, and those triglycerides get packaged together by the Golgi apparatus into this much larger structure called a chylomicron. And chylomicrons exit the cell by exocytosis because they're so big. And these chylomicrons are too big to get into the blood, and instead they enter a lymphatic capillary. So your newly absorbed fats and fat-soluble nutrients don't enter the blood right away. Rather, they enter a lacteal. So chylomicrons, again, are a type of lipoprotein, and I'll show you a picture in just a moment, but lipoproteins there are many different kinds, but lipoproteins are, you can think of them as lipid transport pods. Remember, fats and water don't mix, and sometimes we need to move lipids around our body. So rather than allowing free fat globs to float around in our blood, the lipids are packaged into lipoproteins, and this allows them to safely travel in the blood where they won't find each other and merge into bigger and bigger fat droplets and cause an embolism. So they are lipid transport pods. This is the generic structure of a lipoprotein, any lipoprotein, and chylomicrons are one type of lipoprotein. So there's a core and a shell. And the core of a lipoprotein here, this is a small droplet of fats and fat-soluble things. So it's triglyceride. If this is a chylomicron, it's your newly absorbed fats and your fat-soluble nutrients like vitamin A and vitamin E make up this core. And then surrounding that small droplet of lipid is a monolayer of phospholipids. So this is the monolayer of phospholipids, and you remember that phospholipids are amphipathic. So each one of the heads is hydrophilic, and it is happily interacting with the surrounding water molecules. Water molecules are not shown here, but lipoproteins travel in blood, and blood plasma is mostly water. So this lipoprotein is surrounded by water molecules. And then this phospholipid has two fatty acid tails, and these are lipophilic or hydrophobic. They're lipid-loving, water-fearing, and they are happily interacting with the lipids that make up the core of this phospholipid. So the shell is made out of a monolayer of phospholipids and as well as proteins. So these proteins are called apoproteins, and they're part of the shell of a lipoprotein, and they can be thought of as docking proteins. So this lipoprotein is a lipid transport pod. It is a container of lipids, and it has a specific destination. And so this protein, this apoprotein, will help the lipoprotein reach its specified destination. That protein will interact with some type of receptor. So these are the different lipoproteins in the body, and we can compare their size and their density. Chylomicrons, as you can see here, are enormous relative to the other types of lipoproteins, and chylomicrons carry newly absorbed lipids and lipid-soluble materials. The others, VLDL, IDL, LDL, HDL, they all play a role in transporting lipids from point A to point B, where point A and point B is different for each of these different types of lipoproteins. Maybe you recognize LDL and HDL. Maybe that sounds familiar because you've had a cholesterol panel. So HDL and LDL, maybe you know the good cholesterol versus the bad cholesterol. Cholesterol is a lipid, and it's transported around the body in these two types of lipoproteins. This is a fantastic image. It conveys a lot of important information in a relatively simple format. So what we can see here is a single intestinal villus, and we see in the core of that villus, we see blood capillaries and we see a lacteal. And absorption is occurring, and we know absorption is occurring because in the blood vessels, we see the water-soluble nutrients that were recently absorbed. We see the carbohydrates and the amino acids. We also see small fatty acids because they're little and they can easily get into a blood capillary. And then in the lacteal, we see our chylomicrons, and chylomicrons are newly absorbed. They are lipoproteins that contain newly absorbed lipids and lipid-soluble nutrients. And so the next thing we can do is trace the trajectory for both blood and lymph. So what's the next stop for the blood that contains newly absorbed water-soluble nutrients? And let's take a look at that blood vessel. So these blood capillaries that are in the lacteal, they're going to merge to form venules, and venules to form veins. And this blood is going to travel from the small intestine directly to the liver in a blood vessel known as the hepatic portal vein. And we'll talk more about the hepatic portal system in just a little bit. But for now, you can see that blood that has passed through the small intestine travels immediately to the liver. So the very first stop for these newly absorbed water-soluble nutrients is the liver. And then from there, the blood will go back toward the heart as part of venous circulation. We can also trace the trajectory or the pathway of lymph that includes these newly absorbed chylomicrons. So these newly absorbed lipids that are packaged as lipoproteins, these lipids travel through lymph, and I hope you recall that lymphatic vessels drain into each of the subclavian veins. So these chylomicrons, these newly absorbed fats and fat-soluble nutrients, get dumped into blood without going through the liver first. So once they're absorbed, where do the nutrients go? It depends on if we're talking about water-soluble or fat-soluble nutrients. So water-soluble nutrients are transported through the hepatic portal system directly to the liver, and the liver will remove many of those nutrients from blood, and they can store some of those nutrients, they can package some of those nutrients for later. And if we're talking about fat-soluble nutrients, those chylomicrons are part of lymph, and that lymph does not pass through the liver necessarily, but it gets filtered by a series of lymph nodes, just like all lymph does. And that lymph will become part of blood plasma as the lymphatic system drains into the subclavian veins. Here we see a schematic diagram of blood flow through the hepatic portal system. So blood that's passed through the intestines and other regions of the GI tract, that blood does not immediately go back to the heart, but rather that blood travels through the hepatic portal vein to the liver. And as we can see here, this liver receives both oxygen-rich and oxygen-poor blood. This oxygen-rich blood coming through the hepatic artery, this has recently been ejected from the left ventricle through the aorta and onward. So the hepatic artery delivers oxygen-rich blood to the liver. And then the hepatic portal vein has oxygen-poor blood because this blood has already been through a systemic capillary bed in the GI tract. So it's oxygen-poor, but if the person has eaten recently, it's nutrient-rich. And so this blood is delivered directly to the liver. The liver receives blood from two sources, from the hepatic artery and from the hepatic portal vein. And then after that blood circulates through the liver, we'll take a look at that in just a moment, that blood makes its way back to the heart through regular venous circulation here. So a portal system is a unique circulatory route. So let me start by reminding you of the typical circulatory route that we covered as part of the cardiovascular system. So oxygen-rich blood will leave the heart, it'll leave the left ventricle, it'll travel through a series of arteries away from the heart. Those arteries branch into smaller arterioles, and those arterioles give rise to even smaller, more numerous capillaries. Blood flows through the capillary bed into venules. Venules merge together to form veins, and veins deliver that blood back to the heart. So that's our typical circulatory route. In a portal system, there are two different capillary beds, capillary bed number 1 and capillary bed number 2. And those two capillary beds are connected by what's known as a portal vein. So blood moves from the heart into an artery, into arterioles, into the first capillary bed. In this case, we're talking about the capillary beds in the GI tract. And then through a portal vein, in this case, it's the hepatic portal vein. And then through capillary bed number 2 In that case, those are the capillary beds that occur in the liver. They're called liver sinusoids. And then from capillary bed number 2, we follow the venous route back to the heart. That's a portal system. So this is an artistic representation of the hepatic portal system. So this is blood coming from, let's say, the superior mesenteric artery, if you remember that. And this blood enters capillary bed number 1. And that's a capillary bed that's part of the GI tract. It's passing through a capillary bed in a villus. And that blood takes up the recently absorbed nutrients, the water-soluble nutrients. And then that blood travels not directly back to the heart, but rather it travels through the hepatic portal vein and into capillary bed number 2. And this capillary bed is in the liver. They're liver sinusoids. I'll show you a picture of that in just a moment. And as that blood passes through the liver, 2 things occur. Some of the nutrients get taken up by liver cells because our liver can store certain nutrients. And our liver cells also make a great variety of enzymes that can detoxify potential poisons or toxins that we've taken in. Remember, for most of human evolutionary history, there wasn't agriculture and we were hunter-gatherers. And all organisms make some type of protective chemical. And so lots of plants and fungi, for example, make poisons to protect themselves, to prevent them from being eaten by mammals. And should we ingest a poison, our livers can often modify that substance so that it's no longer toxic to us. So our livers take up some of the newly absorbed nutrients for storage and detoxifies any potential poisons that we have ingested. So after we pass through capillary bed number 2, then we rejoin regular venous circulation, and that blood goes right to the heart. So we have a unique circumstance for blood flow through the liver. It's the only place in our body where oxygen-rich and oxygen-poor blood are combined. And so the graphic artist here cleverly combined red and blue into purple. And you'll recall that red is often used to designate oxygen-rich blood, and we have oxygen-rich blood arriving at the liver through the hepatic artery. And then blue is commonly used to denote oxygen-poor blood. We have oxygen-poor blood arriving at the liver from the hepatic portal vein. It's oxygen-poor but nutrient-rich. And so those 2 sources of blood combine and mix as that blood passes through liver sinusoids. A sinusoid is a giant, super leaky capillary. And we find sinusoids in the liver. And the reason they're so leaky is that in the liver, lots of proteins need to cross the capillary wall. So your liver makes blood proteins, and so those blood proteins need to get into the capillary. Your liver also includes lots of macrophages. They're called Kupffer cells, and these macrophages are capable of taking up cellular debris, microbes that might have accidentally crossed the wall of our GI tract and gotten into our blood. The macrophages are able to capture them. And so as that blood passes through the liver, these liver sinusoids, it's then returned to general circulation. This is venous circulation back to the heart. Here we're looking at a diagram of liver tissue where we have this mixing of oxygen-rich and oxygen-poor blood here. So we have branches from the hepatic artery and branches from the hepatic portal vein. And so these sinusoids eventually merge here, right? So we have oxygen-rich blood mixing with oxygen-poor blood. And as the blood travels through this sinusoid, it's passing through these plates of hepatocytes. And these hepatocytes are interspersed with Kupffer cells, or the macrophages. And so as that blood passes through the sinusoid, these liver cells are removing some of the nutrients for storage, and they're detoxifying potential toxins that we've eaten. And then the Kupffer cells, the macrophages, are collecting any cellular debris or microbes that may have gotten into our blood. All right, so as this nutrient-rich blood passes through our liver, drugs can be metabolized, toxins can be broken down, the microbes can be filtered out and destroyed by those macrophages, and then water-soluble nutrients like amino acids and glucose and water-soluble vitamins and minerals, some of them can be removed by the liver, and the liver can use them or the liver can put them into storage. And the nutrients that stay in the blood and go into general circulation are available to all of our body cells. So we've arrived at the large intestine. The last region of the small intestine is called the ileum, and the first region of the large intestine is called the cecum. So chyme leaves the ileum and moves into the cecum. And once we're in the large intestine, we see several types of propulsion. Two of them are shown here. Propulsion refers to, well, what's going on with the muscularis in order to move the contents of the tube downstream or toward the rectum. So the first type of propulsion we see here is called the gastroileal reflex, and it's a reflex. It's an enteric nervous system reflex, meaning there's a stimulus and then there's a response. And gastro means stomach and ileal means ileum. So this gastroileal reflex is triggered by the arrival of food in the stomach. So distension of the stomach wall and a change in the pH of the stomach is those are the two stimuli that can then lead to the movement of material from the ileum into the cecum. It's almost like the digestive system's way of making more room because she ate her next meal. Okay, everybody move down there. We need more room. She's eating again, more room. And so that chyme is moved into the cecum. So it moves from the small intestine into the large intestine. And then our second type of movement here is called haustral churning. I'll show you an image of the large intestine in just a little bit, but haustra are these pouches that occur along the length of the large intestine. And during haustral churning, a pouch will distend and swell as it fills with chyme. And then that pouch, the muscularis of that pouch, will compress and squeeze and push that chyme into the next haustrum. So that chyme will move from haustrum to haustrum in the direction of the rectum. And that's called haustral churning. Here we see a diagram of the large intestine. So this is what we were talking about just a moment ago. During the gastroileal reflex, an increase in peristalsis in the ileum pushes the chyme through this ileocecal sphincter and into the cecum. The cecum is the first region of the large intestine. And then each one of these pouches is called a haustrum. And during haustral churning, the chyme is forced into a single pouch, and then the walls of that pouch contract and squeeze that chyme into the next pouch. And that's known as haustral churning. These are the three major functions of the large intestine. A little bit of absorption happens in the large intestine. Remember, the vast majority of absorption happens in the small intestine. But in the large intestine, we absorb a little bit of water, and that water absorption makes the difference between liquid and semi-solid chyme. So that little bit of water absorption produces this semi-solid feces. And then some vitamins and minerals are also absorbed in the large intestine. The large intestine also houses what's known as our gut flora, hundreds of species of bacteria that help keep us healthy. They prevent harmful species of bacteria from being able to colonize our large intestines. And some of them also make certain nutrients for us. So we have this mutualistic relationship. They help keep us healthy, and we give them a warm, stable environment in which to live, and they also feed off of our nutrients. These bacteria also convert bilirubin into stercobilin. And we talked about that in the cardiovascular system as part of hemoglobin recycling. And I hope you'll recall that bilirubin is a breakdown product of hemoglobin. And in the large intestine, bilirubin, which arrived there from bile, that bilirubin gets converted into stercobilin, and stercobilin is brown in color. So this is why you can eat bananas and mangoes and avocado and spinach and everything comes out brown because it contains stercobilin. And then the third function of the large intestine is to store feces until defecation. Here are more types of propulsion in the large intestine. So mass movement is also known as mass peristalsis, and this is a super intense, strong, fast peristaltic wave that starts about the center of the transverse colon, and it very quickly drives the contents of the tube toward the rectum. And that will often trigger the defecation reflex, that first sensation that you feel, oh, gotta go, where you go and look for a bathroom. That can be triggered by mass movement, as well as the gastrocolic reflex. So the gastrocolic reflex is a reflex, and meaning there's a particular stimulus and it gives rise to a particular response. So gastro means stomach, colic means colon. And so the stimulus occurs in the stomach when more food arrives in the stomach. The pH of the stomach rises and the wall of the stomach stretches, and that stretching of the wall and the higher pH, those are the stimuli that trigger an enteric nervous system reflex called the gastrocolic reflex, and the motor response is mass peristalsis in the large intestine. So this is another way for the GI tract to say, make more room, she's eating again, everybody move downstream. Only this one happens in the large intestine. The defecation reflex is a spinal reflex, and I hope you recall that means that integration occurs in the spinal cord. And the defecation reflex is triggered when the walls of the rectum fill with feces and the walls stretch. So it's that stretching of the wall of the rectum that's the sensory input that leads to the defecation reflex. And remember, the gastrocolic reflex can trigger mass movement, and that can cause the walls of the rectum to stretch, leading us to the defecation reflex. So in order to defecate, to eliminate the contents of the large intestine, several things need to happen. Number one, the walls of the rectum need to contract, so the lumen of the rectum narrows, and feces are going to move from an area of high pressure to an area of low pressure. So contraction of the wall of the rectum increases the pressure inside the tube, inside the rectum. And then there are two different anal sphincters that both need to relax in order to allow the feces to pass through. Here we see the rectum and the anal canal, and then we have our two sphincters. Remember, the urinary system, between the urinary bladder and the external urethral orifice, the opening of the urethra, we also had two sphincters, an internal and an external sphincter. The same situation occurs here in the GI tract, where the internal anal sphincter is part of the tube, part of the muscularis of the GI tract, and it's made out of smooth muscle. And smooth muscle is not under voluntary control, it's under involuntary control. And then we have the external anal sphincter. The external anal sphincter is part of the urogenital diaphragm. It's made out of skeletal muscle, and we do have voluntary control over it. So during the defecation reflex, that internal anal sphincter is going to relax, and you're going to have that sensation of needing to find the restroom. But what if there's no restroom available? What if you're on Muni or in your car? Then that external anal sphincter is not going to open, and we can voluntarily delay defecation most of the time. And under that circumstance, if we experience the defecation reflex but we don't go to the bathroom because maybe we don't find a bathroom, the contents of the rectum will be pushed back up into the descending colon. But if we do find a restroom, then the external anal sphincter will open and evacuation will occur. So because we are awake, curious human beings, we know that feces vary in consistency. They're not all the same. And the major driver for that change in consistency is the water content of the feces. And we've all had diarrhea, and diarrhea is defined as the defecation of liquidy feces. And the reason why there's so much water in the feces is because the chyme passed through the GI tract too quickly. That's called increased motility of the GI tract. So all of those propulsion mechanisms happen too fast, and as that chyme passes through the GI tract quickly, there's insufficient time for the absorption of water as well as nutrients. And so the end result is diarrhea. So what causes the chyme to pass through the GI tract too quickly? What causes that increase in GI motility? Lots of things. Emotional stress can cause diarrhea, lactose intolerance, as we talked about recently, and microbes that irritate the lining of your GI tract. Food poisoning, as we know, can cause diarrhea. If we're going to talk about diarrhea, we need to talk about its opposite, constipation. Constipation is defined as infrequent defecation. And the ultimate cause of constipation is a decrease in a decrease in GI motility. So the muscularis of the GI tract is propelling chyme too slowly. And when that chyme passes more slowly through the GI tract, there's more time for absorption. Specifically in the large intestine, that's going to lead to the absorption of additional water. So the longer the chyme and then the fecal material, the more time it spends in the large intestine, the more water gets absorbed. And that leads to a dry, hard feces. And that's difficult to pass. And so it can be a self-reinforcing cycle here. As the feces become drier and harder and more difficult to pass, they stay in the large intestine and get even drier and harder and even more difficult to pass. So what causes constipation? Well, ultimately, it's a decrease in the motility of the GI tract, but what leads to that? Dehydration, emotional stress, and yes, emotional stress causes diarrhea in some people and constipation in other people. A lack of dietary fiber. So if you recall, dietary fiber is abundant in plant material. It's the cellulose and hemicellulose that makes up cell walls and seed coats and things like that in plants. And we don't have any digestive enzymes to chemically digest cellulose. So that cellulose remains intact the entire length of the tube. It's not chemically altered at all. And it acts as a bulking agent. So it increases the volume of material in the GI tract, and also certain types of fiber can draw water molecules around them. And so fiber keeps material moving easily along the large intestine. And then famously, certain drugs cause constipation, the opiates. And when opiates are prescribed for pain relief, say following a surgical procedure, sometimes a stool softener is also prescribed, and that stool softener taken along with the opiates can help mitigate some of the constipating effects of Vicodin, for example. And then I see this little symbol in the bottom left corner, and that's that book that I mentioned earlier, What's Your Poo Telling You? And it's a reminder for me to tell you about the entry called Pooless Joe Jackson. And Pooless Joe Jackson was a 12-year-old boy who died. And he was described by his family as defecating very, very infrequently. So he had extreme constipation. And in the months leading up to his death, his stomach became distended. So he had what might be described as like a pot belly. And he's a 12-year-old boy. And he died, and following his death, it was discovered that he has what's now known as Hirschsprung's disease. And Hirschsprung's disease is caused by a problem in the motor neurons and the muscularis of the large intestine. So the motor neurons don't stimulate the muscularis of the large intestine. And so that stimulation, that propulsion of the large intestine is required for defecation. And so poor Joe Jackson didn't defecate as often as he should, and he continued to eat, and so the fecal material accumulated in his large intestine and caused the large intestine to distend and stretch as this fecal material accumulated over time, and eventually the large intestine split, it perforated. And so that perforated large intestine allows the bacteria to now enter the peritoneal cavity. So those bacteria that are part of our normal healthy gut flora, they're absolutely fine and good when they're inside your intestine, but if they enter your abdominal pelvic cavity, they can cause infection, which leads to sepsis, which leads to death. So poor Poolish Joe Jackson died of Hirschsprung's disease when he was about 12 years old. This brings us to the end of the second segment of the digestive system. In the third segment, we'll talk about the phases of digestion, and that'll allow us to zoom out and sort of do an overview of what we've covered so far while learning about the different phases of digestion. See you there.